Hi, my name is Tyler Simpson and I would like to welcome you to the 7th episode of HistoHelpers. Today we will be looking at nervous tissue. Our objectives for today will be to distinguish nervous tissue from other types of tissue, recognize peripheral and central nervous tissue, differentiate white and gray matter, and identify axons and dendrites. We are now looking at slide 58, which is a section of spinal cord prepared with Cresol Violet Luxol Fast Blue stain. The H-shaped gray matter can be seen here at the pointer. In the spinal cord, the gray matter lies deep to the white matter. The white matter can be seen here at the pointer. The centrally located gray matter contains the cell bodies of neurons. The gray matter can be divided into several regions, including the dorsal horn and the ventral horn. The red arrows indicate the dorsal horn, and the green arrows indicate the ventral horn. We are now looking at the dorsal horn using the 20X objective. The dorsal horn contains the cell bodies of sensory or afferent neurons. The yellow arrows point to sensory neurons located within the dorsal horn. We are now looking at the ventral horn using the 20X objective. The ventral horn contains the cell bodies of motor or efferent neurons. A multipolar motor neuron can be seen here circled in green. Nissl bodies, which are aggregates of RAR stained by Cresol Violet, can be seen at the yellow arrow. Multipolar motor neurons contain a large nucleus and a dense nucleolus. The nucleus and nucleolus can also be seen at the blue and red arrows respectively. We have now moved to a different section of gray matter. The gray matter of the spinal cord also contains oligodendrocytes, astrocytes, and glial cells. The red arrows point to several oligodendrocyte nuclei. Note that oligodendrocytes have characteristic small, dark, and round nuclei. Oligodendrocytes often have a clear zone surrounding the nucleus, or a perinuclear halo, which is an artifact of fixation. The yellow arrows indicate several astrocytes. Astrocytes can be distinguished by their large, ovoid nuclei, which are often lightly stained. We have now moved to a different section of the slide to examine the white matter of the spinal cord. The white matter of the spinal cord is peripherally located and it does not contain nerve cell bodies. The white matter is primarily comprised of myelinated axons and the cell bodies of oligodendrocytes. The white matter can be seen here at the pointer. Zooming into the 20X objective, an oligodendrocyte can be seen at the green arrow, with its myelin sheath surrounding a nearby axon. We have now moved to the periphery of the spinal cord to examine the meninges. The spinal cord itself is invested in the meninges, which function as a three-layered protective coating. The dura mater is the outermost fibrous layer. The dura mater is not seen in this section. The arachnoid mater is the middle layer and can be seen at the green arrow. The subarachnoid space, which is deep to the arachnoid mater, can be seen at the yellow arrow. The pia mater is the innermost layer and is closely adhered to the white matter of the spinal cord. The pia mater can be seen at the red arrow. We are now looking at slide 211, which is a section of cerebellum prepared with H&E. The arrangement of gray and white matter in the cerebellum is opposite of that which is seen in the spinal cord. The cerebellum consists of a core of white matter, which is surrounded by the superficially located gray matter. The white matter can be seen at the green arrow. The gray matter can be seen at the blue arrows. We have now zoomed in on the gray matter. The gray matter can be subdivided into three layers: the outer molecular layer, the middle Purkinje cell layer, and the inner granular layer. These three layers can collectively be referred to as the cerebellar cortex. The molecular layer can be seen spanning the red line. The Purkinje cell layer can be seen at the blue arrows. The granular layer can be seen spanning the green line. Notice that the molecular layer appears much lighter than the granular layer. The darker appearance of the granular layer occurs due to the great number of darkly staining nuclei packed closely together. A portion of the white matter can also be seen spanning the yellow line. We are now looking at slide 210, which is a section of cerebrum prepared with H&E. In both the cerebrum and the cerebellum, the arrangement of gray and white matter is opposite of that which is seen in the spinal cord. The gray matter and the white matter can be seen at the green and yellow arrows respectively. The gray matter of the cerebrum contains 6 visible layers and is comprised of neurons and glia. The white matter of the cerebrum is comprised primarily of axons. We have now zoomed in on the gray matter. Large pyramidal cells can be found in the gray matter. Pyramidal cells contain apical dendrites that point towards the external surface of the cerebrum. A pyramidal cell is circled here in green. The blue arrow points to the apical dendrite. Note that only the proximal portions of the apical dendrite can be seen because H&E stains only the Nissl bodies of the cytoplasm, which do not extend beyond the proximal segment of the apical dendrites. Axonal processes can also be seen extending from the base of this pyramidal cell. We are now looking at slide 53, which is a section of cerebellum prepared with silver stain. Recall that the cerebellar cortex is composed of the outer molecular layer, the middle Purkinje cell layer, and the inner granular layer. Using the 20x objective, a Purkinje cell can be seen at the green arrow. Purkinje cells are globular-shaped cells that send their axons into the granular layer. The dendrites of Purkinje cells arborize in the molecular layer. Neuronal axons are stained in black and can be seen synapsing on Purkinje cells. We are now looking at slide 63, which is a section of cerebrum prepared with gold chloride stain. This section of cerebrum has a matted appearance due to the interweaving nerve cell and glial cell processes. Zooming into the 40x objective, an astrocyte can be seen at the tip of the blue arrows. Fibrous astrocytes contain long processes that extend towards blood vessels. Astrocytic processes form an astrocyte-endothelial cell interaction, which induces the endothelial cells to form the blood-brain barrier. Fibrous astrocytes are found primarily in the white matter and contain long, slender processes with numerous filaments. Protoplasmic astrocytes are found primarily in the gray matter and contain thick, bushy processes. We are now looking at slide 72, which is a section of dorsal root ganglion prepared with H&E. The dorsal root ganglion is a sensory ganglion. The DRG is seen here circled in blue. Recall that the DRG houses the cell bodies of sensory neurons. Using the 20x objective, the neuronal soma with its surrounding satellite cells can be seen. A large round neuronal soma is circled here in green. The cell bodies usually contain round and centrally located nuclei and nucleoli, which can be seen at the blue and orange arrows respectively. Satellite cells can be seen surrounding the neuronal cell body at the red arrow. Nissl bodies can be seen within the cytoplasm of neurons. Recall that Nissl bodies are aggregates of RER. Lipofuscin is a brownish gold pigment that is visible in routine H&E preparation. Lipofuscin can be seen at the yellow arrow. Lipofuscin accumulates in long-lived eukaryotic cells as a result of cellular senescence, or aging. Thus, it is often called the wear and tear pigment. It is a conglomerate of oxidized lipids, phospholipids, metals, and organic molecules that accumulate within the cell as a result of oxidative degradation of mitochondria and lysosomal digestion. We are now looking at slide 215, which is a section of a peripheral nerve prepared with H&E. This slide contains both the cross-section and the longitudinal section of a peripheral nerve. In the cross-section on the left, the epineurium can be seen at the red arrow. The epineurium surrounds multiple fascicles. The perineurium can be seen at the blue arrow. The perineurium surrounds a single fascicle, which consists of bundles of axons. Using the 40x objective, the endoneurium can be seen at the yellow arrow. The endoneurium surrounds individual nerve axons. An axon can be seen at the green arrow with its surrounding myelin sheath at the black arrow. We have now moved to the longitudinal section where the peripheral nerves appear to follow more of a zigzag course. Schwann cells and fibroblasts can be seen. Schwann cells can be distinguished by their pale stained nuclei. The yellow arrow points to the nuclei of a Schwann cell. Fibroblast nuclei are thin and dark. The red arrow points to the nuclei of a fibroblast. Axons can also be seen surrounded by their myelin sheaths, except where the nodes of Ranvier are located. The black arrow points to an axon. Its myelin sheath can be seen at the blue arrow. A node of Ranvier can be seen at the green arrow. That's a wrap for episode 7. Tune in next time for a heart-pounding look at the circulatory system. Thanks for watching HistoHelpers. We're here to help.